some questions regarding.
I'm just wondering, do the perturbations, is your idea to introduce these perturbations, does that only work if the contestants don't have that access to the perturbations? Would the perturbations be part of your training set? Or does it specifically have to be something on the validation person's end that wasn't accessible to your memory first?
Okay, so I'm not sure I completely understand the question, but if I interpret it correctly, I think it would have to be something that the competition organizers would need to introduce because the model that Jonathan Carr built is very extensive, and I think it'd be very cumbersome for participants to go in and change that to add their own perturbations. So I haven't given it too much thought about how it would actually be implemented, but I just think that would be a cool idea.
I have a question to Clemens. So at the end of your presentation, you made a remark about biological knowledge versus just statistical thinking. And I wasn't sure which way you were going with that comment, because as a biologist and a practitioner, I don't think people should just be cranking data for the data's sake. I mean, there has to be some sort of understanding what the underlying problem is. And then people have to be thinking what other data or information may be useful, because then the solution is not only right, but is also one that can be communicated back to biologists. So can you expand more on your final comment?
So I think you have to decide either you want to assess the current methodology, the statistical methodology which is applied in a field, and then you have to make a challenge which somehow is appropriate for assessing this. If you are interested in learning something about a biological problem, and if you want to that many people, many groups work on it, and that maybe at the end you have learned a lot, then of course it's another purpose. Then it's not the purpose of assessing the methods, then it's more working on a problem. Yeah.
I have a question for Alex, actually. Why didn't you use the perturbation data, given that, I mean, I know you wanted the perturbation in the middle, but,
yes.
and in the middle of the simulation, not as we did at the beginning, but
yeah.
It seemed that maybe that would have been useful for you.
Yes. So that was one of the things that I was going to mention in the talk, but sort of cut out for time purposes. I think it did occur to me that it would be useful to buy the perturbation data from the competition. But I think the thing that was the biggest challenge in that is you would have to buy the perturbation data. Then you would have to simulate your candidate models, not just for their original parameters, but then re-simulate them under the perturbed condition. So now you're going from 24 hours of compute time to 48. And then just doing one perturbation might not be enough, so you'd have to do several perturbations. So I think that approach could work given more computational resources, but it might be the best in terms of computational efficiency if as many perturbations could be incorporated in a short time period as possible. So perturbing the system at multiple points along a single simulation run. That to me seems like it would be more useful.
One question for the team cracks. But we'll discuss about this, what do we mean by a method, if it is a method in the abstract or is a method to solve a biological problem. I think that when we say DREAM is assessment of methods, are methods to solve a biological problem, not a method in the abstract. But we'll discuss that later because I think it's an interesting discussion. I wanted to ask you, because I couldn't really abstract it from your presentation. I was a little distracted. But I want you to explain a little bit more whether, the extent to which you use the distance, the distance to the real parameters, because that's, in a way, whether, how you use that, and whether, you were thinking while using that would never be a datum that you have from a real experiment, right? So why would you use that? But maybe I didn't understand how you used it. So first the question is how you used it. And if you used it explicitly, as a piece of information to guide your search of parameters, then why you used it? Because you know that that is something that you would never get in a real experiment.
So if you want to estimate the parameters, then you have to exploit the whole information you get from your experimental data somehow, yeah? And of course, you can see it as an optimization problem, which somehow was simplified by having this somehow perfect measurement. And this, of course, is exactly the reason why I think if you have something like a leaderboard, then you have something like perfect information about your system. And in biology, in scientific applications, you never have this kind of perfect information. You don't know how far you are from the reality, from the truth. And this is the reason why I think providing such information in a challenge somehow violates the natural setting you have in applications. And
you know, the reason why it was a mistake it was our mistake to give that in every single run. The idea was to give it only, because the challenge was so difficult, to give it only in one instance of the leaderboard, so that you have a sense. So that what I don't understand very well, you know, is if you use it consistently, then in a way you are solving a problem very different from the problem that we were trying to solve. So we didn't learn much from that, even though you solved the problem. It's like we will know how to use it in real life. That's my, you know,
yes, I mean,
I'm sad about that.
Yes, of course. This is the reason why I think in general one should not give this information to participants, right? I mean, also if you have a score once, it's very informative. Yeah, it's. Yeah.
So,
please.
hello. Okay. So, just commenting on the question that was just asked. In a lot of cases, you have, for instance, a small amount of experimental data available. And that's typically the case where you're looking at expression data. Everyone has expression data. Not everyone has metabolomics data. So in some cases, you can develop a method that tries to perform well by just looking at this smaller segment of the data set. But you can still measure it against some larger standards. So it's maybe not totally unrealistic to have something to compare against, to be able to compare against the actual parameters in the model. And related to that, I was just wondering, as far as the random forest method goes, I'm not an expert in these methods, but is it possible that you could select several solutions from the random forest model before it converges on one particular solution? And because I noticed that when you showed your parameters, they were all really close together compared to the So if you had it to do over again, could you pick several of those and compare to the parameters that were
you can. It's one of the few parameters that you can tune in the algorithm. You can grow 1,000 trees and then average them out, or you can grow 20 trees and average them. And then if you the fewer trees you grow, the more stochastic your result is going to be, and that will create a larger spread. So, I mean, the way that I ended up doing it, I don't know if this was the best way, was I did as many trees until it started to converge, and then that gave a pretty consistent result. And then what I did was randomized and did a local search around that point. So sort of added the variability randomly, but maybe doing it the way that you described would work better. I'm not sure.
Okay, thanks.
Okay, thanks a lot.